Hello, in today's video, we shall learn in detail about mechanism of photosynthesis in higher plants. By the end of this video, you will have a clear understanding about photosystem 1 and photosystem 2, cyclic and non-cyclic photophosphorylation, light and dark reactions, Kelvin cycle, Hertzsprung pathway, differences between C3 and C4 plants, and the factors affecting the rate of photosynthesis. Let us start with the introduction to photosynthesis. Photosynthesis is defined as an intracellular, physico-chemical, anabolic, endergonic process by which green parts of the plants synthesize carbohydrates in the presence of sunlight, chlorophyll, carbon dioxide, and water. Oxygen is given out as a byproduct. Chloroplasts are the sites of photosynthesis. Pigments involved in photosynthesis are chlorophylls, carotenes, and xanthophylls. Chlorophyll A and chlorophyll B show maximum absorption in red and blue regions of visible light. The pigments are organized into two light harvesting complexes with photosystem 1 and photosystem 2. There are two forms of chlorophyll A, P700 and P680, which form reaction centers of photosystem 1 and photosystem 2 respectively for the photochemical reaction. Mechanism of photosynthesis involves two distinct phases: light-dependent phase or light reaction and light-independent phase or dark reaction, otherwise known as Kelvin cycle. Let us learn about the structure of chloroplast, which is the site of photosynthesis. In plants, photosynthesis takes place in chloroplast, which contain chlorophyll pigments. Chloroplast is a lens-shaped organelle bounded by a double membrane called peristomium, which is made up of lipoproteins. It encloses colorless colloidal matrix called stroma. Embedded in the stroma are the grana, which are made up of stacks of disc-shaped structures known as thylakoids. Grana of the chloroplast contain the chlorophyll pigment, while the stroma contain DNA, ribosomes, RNA, and various enzymes needed for the dark reaction of photosynthesis. DNA is in the form of a closed circular, highly folded naked ring and is termed as plastidome. As chloroplast contains DNA and ribosomes, it is self-replicating and semi-autonomous organelle. Grana are interconnected by membranes called stroma lamellae, which help in rapid transport of materials. The grana are the site of light reaction of photosynthesis. It stores energy in the form of adenosine triphosphate ATP and nicotinamide adenine dinucleotide hydrogen phosphate NADPH2 and is responsible for photolysis of water. Dark reaction occurs in the stroma. Present along the inner surface of grana lamellae are the photosynthetic units or quantosomes. Each quantosome contains a number of chlorophyll molecules. It forms two photosystems photosystem 1 and photosystem 2. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Let us understand about the different photosynthetic pigments. Pigments are substances that have the ability to absorb light at specific wavelengths. In higher plants, chlorophyll A, chlorophyll B, carotenes and xanthophylls are main photosynthetic pigments. Chlorophyll A is the only pigment that can absorb and convert light energy into chemical energy. Hence, it is called essential pigment and it acts as reaction center. Each photosynthetic pigment has a set of wavelength that it absorbs called as absorption spectrum. It is a curve or graphic representation of the amount of different wavelengths of light absorbed by a pigment. Absorption spectra can be depicted by wavelength on the x-axis and the degree of light absorption on the y-axis. Maximum absorption of visible light by photosynthetic pigment occurs in red and blue region and little absorption in yellow and orange region of spectrum. Action spectrum is a curve or graph representing the overall rate of photosynthesis at different wavelengths of light. The rate of photosynthesis is measured in terms of oxygen evolved and carbon dioxide utilized. The action spectrum shows the highest peak in red region and another smaller peak in blue region. Chlorophyll B, xanthophylls and carotenes are accessory pigments which also absorb light and transfer the energy to chlorophyll A that forms the reaction center. The accessory pigments and the reaction center together form photosystem. Both photosystem 1 and photosystem 2 are required for oxygenic photosynthesis. The pigments are organized into light harvesting complexes within the photosystem 1 and photosystem 2. Each photosystem has two parts, a reaction center and an antenna complex surrounding the reaction center. The antenna complex contains chlorophyll molecules which capture and transfer the light energy to the reaction center. Photosystem 1 absorbs the longer wavelength of light, which is 700 nanometer, while photosystem 2 absorbs the shorter wavelength of light, which is 680 nanometer. Let us study the differences between photosystem 1 and photosystem 2. Photosystem 1 has P700 as the reaction center. Photosystem 2 has P680 as the reaction center. The pigment in photosystem 1 absorbs longer wavelength of light, which is 700 nanometer. The pigment in photosystem 2 absorbs shorter wavelength of light, which is 680 nanometer. Photosystem 1 lies on the outer surface of the thylakoid membrane. Photosystem 2 lies on the inner surface of the thylakoid membrane. Photosystem 1 is involved in cyclic and non-cyclic photophosphorylation. Photosystem 2 is involved only in non-cyclic photophosphorylation. Photosystem 1 is not involved in the photolysis of water. Photosystem 2 is involved in the photolysis of water. Photosystem 1 donates the electrons to ferredoxin reducing substance. Photosystem 2 donates the electrons to quinone. Functions of photosystem 1 are ATP synthesis in cyclic photophosphorylation and NADPH2 synthesis in non-cyclic photophosphorylation. Functions of photosystem 2 are ATP synthesis and photolysis of water. Next are the light-dependent reactions, otherwise called light reactions. It is the first phase of photosynthesis occurring in the grana of chloroplast. Light energy is absorbed by the photosynthetic pigments and is converted into chemical energy in the form of ATP by photophosphorylation. Photolysis of water takes place, leading to the formation of NADPH2 and liberation of oxygen. Photophosphorylation is the process of ATP synthesis from ADP and inorganic phosphate with the help of solar energy and chlorophyll. At reaction center, energy-rich electrons are expelled on receiving photons, which leads to an electron transfer. Dr. Arnon suggested that there are two pathways for this electron transfer. They are cyclic photophosphorylation and non-cyclic photophosphorylation. Cyclic photophosphorylation involves photosystem 1. When light falls on photosystem 1, it gets excited and a pair of energy-rich electrons are emitted by reaction center P700. These electrons are accepted by electron acceptor, namely ferredoxin reducing substance, and are transferred via ferredoxin, cytochrome B6, cytochrome F, and plastocyanin. From plastocyanin, the electrons return back to the P700 chlorophyll molecule. ATP molecule is synthesized when electron is transferred from cytochrome B6 to cytochrome F. Since the electron returns back to the same chlorophyll molecule, it is called cyclic photophosphorylation. Non-cyclic photophosphorylation. Photosystem 2 absorbs light energy which is passed to the reaction center, that is chlorophyll A 680 or P680. The energy-rich electron is emitted from P680 and is accepted by primary acceptor coenzyme quinone. From here, the electrons are transferred to plastoquinone, cytochrome B6, cytochrome F, and plastocyanin. One ATP molecule is synthesized when electron passes from cytochrome B6 to cytochrome F. The reduced plastocyanin transfers the electrons to the reaction center of P700. It releases energy-rich electrons which are accepted by ferredoxin-reducing substance and transferred to nicotinamide adenine dinucleotide phosphate to form negatively charged NADP in the presence of enzyme ferredoxin NADP reductase. P680 brings about splitting of water molecules to release oxygen. Splitting of water molecules into H plus and OH minus ions in presence of light and chlorophyll is known as photolysis of water. The 4 electrons generated during breakdown of 4 water molecules are accepted by 2 NADP to form 2 NADPH2. Therefore, non-cyclic photophosphorylation is concerned with photolysis of water, formation of ATP and NADPH2 molecules. ATP and NADPH2 are together known as assimilatory power. Let us look into the differences between cyclic and non-cyclic photophosphorylation. Cyclic photophosphorylation is performed by photosystem 1. In non-cyclic photophosphorylation, both photosystem 1 and photosystem 2 are involved. In cyclic photophosphorylation, P700 is the active reaction center. In non-cyclic photophosphorylation, P680 is the active reaction center. Electrons flow in a cyclic manner in cyclic photophosphorylation. In non-cyclic photophosphorylation, electrons flow linearly in a zigzag pattern. Electrons in cyclic photophosphorylation return back to photosystem 1. Electrons given out by photosystem 2 does not return back to it, but is used by photosystem 1 in non-cyclic photophosphorylation. In cyclic photophosphorylation, photolysis of water does not take place. In non-cyclic photophosphorylation, photolysis of water takes place. Oxygen is not evolved in cyclic photophosphorylation. In non-cyclic photophosphorylation, oxygen is liberated due to photolysis of water. In cyclic photophosphorylation, the final electron acceptor is photosystem 1. In non-cyclic photophosphorylation, the final electron acceptor is NADP. Cyclic photophosphorylation occurs in photosynthetic bacteria. Non-cyclic photophosphorylation primarily occurs in higher plants. Next, let us try to understand chemiosmotic hypothesis or mechanism of ATP synthesis. Chemiosmosis is the movement of ions across a selectively permeable membrane. It is a biological process proposed by P.D. Mitchell. Splitting of water molecules on the inner side of the thylakoid membrane results in H plus ion accumulation within the lumen of thylakoids. An electrochemical concentration gradient of protons develop when H plus ions or protons diffuse from an area of higher proton concentration to an area of lower proton concentration. ATP synthetase allows protons to pass through the membrane. In the stroma side of the membrane, NADP reductase enzyme is present. Protons are required for the reduction of NADP to NADPH2. Thus, number of protons decreases in the stroma and the number of protons increases in the lumen. Due to the proton gradient across the thylakoid membrane, there is a rapid movement of protons generating energy which is used for the synthesis of ATP. Next are the light-independent reactions or dark reactions. This is the second phase of photosynthesis, which occurs in the stroma of chloroplast. It is independent of light and hence known as dark reaction. It is a biochemical reaction in which CO2 is reduced to form carbohydrates with the help of ATP and NADPH2. Kelvin worked out the pathway of carbon, and therefore light-independent reactions or dark reactions are also known as Kelvin cycle. Since the first stable product formed is a 3-carbon compound, it is also known as C3 pathway. Kelvin cycle can be divided into 3 phases: carboxylation phase, reduction phase, and regeneration phase. Carboxylation phase or carbon fixation. A 5-carbon compound, ribulose 1,5-bisphosphate RuBP, reacts with atmospheric carbon dioxide in the presence of enzyme RuBP carboxylase, otherwise known as Rubisco, to form an unstable 6-carbon compound, which immediately splits by hydrolysis into 2 molecules of 3-carbon compound called phosphoglyceric acid. Reduction phase. The phosphoglyceric acid in the presence of ATP produces 1,3-diphosphoglyceric acid, which is reduced to phosphoglyceraldehyde by using NADPH2. In the presence of enzyme triose phosphate isomerase, some molecules of 3-PGAL are converted into its isomer dihydroxyacetone phosphate DHAP. Next, synthesis of sugar and regeneration. 6 molecules of RuBP and 6 molecules of carbon dioxide are required for the synthesis of 1 molecule of glucose. Out of 12 molecules of PGAL, 2 molecules are used for the synthesis of glucose. A molecule of PGAL and a molecule of DHAP combine together to form a molecule of fructose 1,6-diphosphate. Fructose 1,6-diphosphate forms fructose 6-phosphate, which in turn forms glucose 6-phosphate, and finally glucose is formed after dephosphorylation. Out of 12 molecules of PGAL, 10 molecules are utilized for the regeneration of 6 molecules of ribulose monophosphate RuMP, which on phosphorylation form RuBP using ATP. Having learnt in detail about C3 pathway, let us take up C4 pathway, which is known as Hatch-Slack pathway. This pathway is seen in certain tropical plants like maize, sugarcane, etc. They can photosynthesize in high temperature and high intensity of light. The first stable compound is a 4-carbon compound, oxaloacetic acid, and hence these plants are called C4 plants. C4 plants have a special type of leaf anatomy known as Kranz anatomy. The mesophyll cells are not differentiated and are homogeneous. The vascular bundles are surrounded by radially arranged large bundle sheath cells. The chloroplasts are dimorphic. The chloroplast in mesophyll cells contain granule chloroplast. They are small in size and more in number. Chloroplast in bundle sheath cells contain agranule chloroplast, that is, chloroplasts are without grana. They are bigger in size but less in number. Let us understand the mechanism of C4 pathway. Reactions in mesophyll chloroplast. First, carbon dioxide fixation or carboxylation. Phosphoenol pyruvic acid present in the chloroplast of the mesophyll cells accepts atmospheric carbon dioxide to form a 4-carbon compound, oxaloacetic acid. Next is reduction. In the presence of NADPH2, oxaloacetic acid is reduced to malic acid by enzyme malate dehydrogenase. Next is reactions in bundle sheath chloroplast. Decarboxylation. Malic acid formed in the mesophyll cells moved to the chloroplast of bundle sheath cells. It undergoes decarboxylation in the presence of NADP and malate dehydrogenase to form pyruvic acid and carbon dioxide is released. Carbon dioxide fixation. The carbon dioxide released is accepted by RUBP and is fixed by C3 pathway or Kelvin cycle. Thus, glucose is formed by Kelvin cycle, which is either transported through phloem or is stored as starch in bundle sheath chloroplast. Pyruvic acid produced from malic acid is transported to mesophyll cells, and with the help of ATP, it undergoes phosphorylation to regenerate PEPA. Let us compare C3 plant and C4 plants. C3 plants use Kelvin cycle for dark reaction of photosynthesis. C4 plants use Hatch-Slack pathway for dark reaction of photosynthesis. In C3 plants, carbon dioxide acceptor is RUBP. In C4 plants, carbon dioxide acceptor is PEPA or phosphoenol pyruvic acid. First stable product in C3 plants during photosynthesis is phosphoglyceric acid. First stable product in C4 plants during photosynthesis is oxaloacetic acid. Leaves of C3 plants do not have Kranz anatomy. Leaves of C4 plants have Kranz anatomy. Carbon dioxide fixation takes place only at one place in C3 plants, that is, in mesophyll cells. Carbon dioxide fixation takes place twice in C4 plants, once in mesophyll cells and then in bundle sheath cells. Bundle sheath cells of C3 plants do not contain chloroplast. Bundle sheath cells of C4 plants contain chloroplast. In C3 plants, chloroplasts are monomorphic, that is, they have only one type of chloroplast. In C4 plants, chloroplasts are dimorphic, granule and agranule chloroplast. Photorespiration rate is very high in C3 plants, which leads to loss of fixed carbon dioxide. In C4 plants, photorespiration is absent. In C3 plants, optimum temperature for photosynthesis is very low. In C4 plants, optimum temperature for photosynthesis is high. In C3 plants, carbon dioxide compensation point is high. In C4 plants, carbon dioxide compensation point is low. C3 plants are less efficient in photosynthesis. C4 plants are more efficient in photosynthesis. C3 plants are abundant in temperate regions. C4 plants are abundant in tropical regions. Examples of C3 plants are potato, paddy, etc. Examples of C4 plants are maize, sugarcane, etc. Finally, we shall take up factors affecting rate of photosynthesis. Photosynthesis is affected by both internal and external factors. Internal factors are number and size of leaves, orientation of leaves, arrangement of mesophyll cells, presence of chloroplast, also amount of chlorophyll present, and many others. External factors are light, carbon dioxide concentration, temperature, and water. Light. Three aspects of light as a factor affecting rate of photosynthesis are light quality, intensity, and duration. Rate of photosynthesis is more in the red and blue regions of light. At very high light intensity, rate of photosynthesis decreases as breakdown of chlorophyll molecule takes place. Carbon dioxide concentration. The major limiting factor for photosynthesis is carbon dioxide. Up to 0.05% of carbon dioxide level in the atmosphere can increase the rate of photosynthesis, but beyond this, it causes harm to the plants. Temperature. Tropical plants have a higher temperature optimum than the temperate plants. C4 plants show higher rate of photosynthesis at higher temperature. Water. Water stress results in closure of stomata, which causes reduction in carbon dioxide availability. Though several factors interact and affect photosynthesis, it is usually one factor that limits the rate of photosynthesis. In 1905, Blackman proposed the law of limiting factors, which states that if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the rate if its quantity is changed. So today we have learnt in detail about mechanism of photosynthesis in higher plants, differences between cyclic and non-cyclic photophosphorylation, C3 and C4 plants, photosystem 1 and photosystem 2, light and dark reactions, Kelvin cycle, Hertzsprung pathway, and also the various factors that affect the rate of photosynthesis. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.